Somebody's having trouble logging in, it looks like. All right, are there any questions? Oh my goodness. Try and get that off of there. Let's see. Just a minute, Jan, let me block you. There we go. All right. A little sorry about that. Does anybody have any questions? If not, let me go ahead and share the screen and begin. Okay, we're covering Lab 01 today, the ubiquity of microorganisms. Let me remind you, you should read the infectious disease research project file called Project Instructions in Canvas for next week. And I think I've got that up here. Give me 1 minute. Well, I thought I had it up there, but I've actually just got the that link. So where is that? Week 1 lab, there it is. Nope, must be under week 2 lab. Sorry, my mouse isn't working. There it goes. Yeah. Is that what you're supposed to be reading? The Infectious Disease Research Project? Yeah, Infectious Disease Research Project. And that's what it's actually called, not Project Instructions. It's the Infectious Disease Research Project. And I'll have to change that. So just delete that right there. All right, let's move on. To Lab 01. Ubiquity of microorganisms. And you should do some reading before you do this lab, 157 to page 162, and then page 162 to 163. There are some video clips throughout this lab module. Let me go ahead and get the exercises out while I'm there. And then come back to here. So the primary learning objectives, upon successfully completing this laboratory, you should be able to know that microorganisms are almost everywhere. You should be able to describe the purpose of culture media. You should be able to describe the colony abundance, the colony diversity, and the colony morphology. And we'll talk a little bit about those. You should be able to define the following terms. What's inoculation? Ubiquity of microorganisms. What's something that's sterile? What is isolation? Incubation, in vitro, culture media. And we have 2 different types. We have a liquid culture, which is a broth, and then a solid culture, which is usually solid from an agar being added to it. And then you can have these cultures in a plate, a slant, or a deep. And I think I'll show you each of those today. You should know what fastidious means, what non-selective media is, what ubiquitous means, and of course, what is a colony. Any question about any of that? Microorganisms or organisms that are too small to be seen with the naked eye. You need at least a light microscope or an electron microscope in order to see them. Viruses and prions are included, even though they are not cells, and most people say they are not alive, although some people could argue that. We're not going to get into that. Microorganisms also include the helminths, which are worms. And I've already discussed that in the lecture. The phrase ubiquity of microorganism refers to the concept that microorganisms are everywhere. In this lab, we will investigate the abundance and diversity of microorganisms in common environments. We will also learn basic lab protocols, including the use of solid media, labeling lab cultures, procedures for disinfecting lab benches, and procedures for washing our hands. You should know that a culture media, also called a growth medium, is a sterile laboratory preparation. Like if you were to get a culture medium in a real microbiology lab, if you were to walk into the lab and pick up a culture media, it will be sterile. And usually how they sterilize it is they sterilize it in an autoclave. And we'll talk a little bit about that, an autoclave, later. Mediums contain a variety of nutrients, and these nutrients are required for growing the microorganisms. Oops, the lecture just converted. Let me go ahead and do this. And figure out where this goes. Should be under documents and Zoom. There it is. Probably right there. That's a little bit new setup, and I'm not used to that coming up, and then I've got to figure out where it's going to save down. All right. The culture medium is also called a growth media, and it is sterilized. Culture medias can be liquid. These are called broths, and they're commonly used in a test tube, liquid broths. You can add agar to a liquid media to get a solid medium, and you can add some other substances like Jello, but most of the time we add agar. There's a little video you should watch on preparing pour plates. I'll let you do that. After I'm done talking, I'm just giving a brief overview of the lesson. You should know that the media we're going to talk about today are general purpose media. There are different types of media, and today we're only going to talk about general purpose media. A general purpose media is a media which is non-selective. It usually is complex in its composition, meaning it has lots of molecules in it, different molecules, and they support the growth of non-fastidious organisms by using peptone, yeast, and beef extracts. It has many molecules in it because usually we just grind up something and make a media out of it, like we grind up a cow and we add beef extracts. And that cow can have many different molecules depending on what the cow had been eating. And that's why we say it's complex. And it grows many different organisms, but an organism that requires specific additives to grow is one that we call fastidious, and they do not grow easily, and they do not grow in a general purpose media. An example is Streptococcus pyogenes is a fastidious organism. It causes strep throat and a whole bunch of other human diseases. I think it can cause necrotizing fasciitis, but it doesn't cause all necrotizing fasciitis. And I don't remember if this is the one that causes like 80% or 20% of necrotizing fasciitis. But my point is it causes many diseases besides strep throat. And today, because of the use of antibiotics whenever somebody gets strep throat or sore throat, some of the diseases that it causes are actually very rare today. Like I think it causes scarlet fever. And that's a disease which is very rare today because most people, when they get sick, they get antibiotics and then the disease doesn't go on to cause scarlet fever. My point is, getting back to my point, Streptococcus pyogenes is a fastidious organism and it will not grow on a general purpose media. You have to add specific ingredients to the media to get Streptococcus pyogenes to grow. And an easy way to get Streptococcus pyogenes to grow is to grow it on an enriched plate, an enriched media plate that has blood in the plate. And the red blood cells will provide the nutrients that Streptococcus pyogenes requires before it will grow. And don't ask me what those nutrients are. I just know if you add red blood cells to the media, you can get Streptococcus pyogenes to grow. Any question about any of that? So organisms that can grow on a general purpose media are most things we have in the lab, like E. coli, Staphylococcus aureus, Staphylococcus epidermidis, Micrococcus luteus. Common organisms we grow in the lab generally grow on a general purpose media. They're non-selective. And general purpose media tend to grow many different bacteria. Most of the bacteria we grow in a microbiology lab can grow on a general purpose media. The general purpose media include things, I'll start from the bottom, nutrient agar, abbreviated NA, tryptic soy agar, abbreviated TSA, and then nutrient agar where we've added yeast extracts to it. And this is a little richer media, the nutrient agar. Like nutrient agar, I think all it has is ground up beef extract, ground up, what is it, soy product? Something like that. It only has 3 additives to it. And I just remember the nutrients mainly come from beef extract. And then this one has nutrient agar and we add yeast extract, ground up yeast. And that one we abbreviate NA, nutrient agar with yeast extract, NA. Any questions about any of that? All right. We label a Petri dish with or a tube to correctly know what's in the tube and whose it is and what date it is. So all tubes and plates should be correctly labeled. You always put your name or your initials. You always put the date. You put the type of medium like Na, which here the media would be TSA. And then the temperature that you're incubating the plate is optional. You can put the incubation temperature, which that isn't on there. And then you put the inoculum, what you're growing in the culture. If you know what it is, state what it is, like this is E. coli, Escherichia coli. And if you don't know what it is, just write down where it came from. Like you don't write down growth from air exposure, you write down something like air exposure. Okay, so you write down your inoculum. You can write a piece of tape, which on test tubes, that's what you need to do, to then write this information On the plate, an agar plate, it's best to write around the perimeter of the base, meaning the edge of the plate. And that way your writing doesn't block what you're trying to view in the plate. Okay. Any questions about that? All things in the lab must be appropriately labeled with those 4 things. An inoculation is the placement or transfer of a microbial specimen into a medium. So when we inoculate a tube with E. coli, when we use the word inoculation, that means putting the E. coli in the tube. Incubation is then taking the inoculated culture media and putting it in the correct place for growing, usually in an incubator, and then allow the culture to grow the microbes. So that's what incubation is. I think I've got a picture of an incubator down here. Yep, there's an incubator. And they can be complex or simple. Most of the incubators in our lab, all they do is they heat the cultures and then provide a little bit of humidity, like there's probably liquid down there, and that provides a little bit of humidity. You can have fancy ones that will make the environment a specific level of oxygen and a specific level of CO2, and they can do some other things as well. All right, so agar plates, if you were to put them in, let me try and view this. I can't see this, so hopefully you can view this. If you're looking at this, we're going to pretend this is an agar plate. If you were to put it in an incubator right side up with the lid on the top and the plate with the growth on it on the bottom, the moisture from the agar will, from the heat of the incubator, evaporate, and then it will condense on the bottom of the lid. I don't know if you can see that. Hopefully you can. I'm not seeing anything, so I can't see what I'm showing you. And then the drop would form, and then it would fall down onto the culture, dispersing the culture. And that could ruin the culture. You don't want to disperse your culture. If the drop were to come down the edge of the plate and then go into the plate, it could actually contaminate the plate. But the water itself, when it forms a droplet on the lid, it will be sterile. So you don't necessarily contaminate the plate, but you do increase the risk of contaminating the plate. So what we do is all plates that we put in the incubator, we put them in upside down. Now, when moisture evaporates out of the agar, it will form, the moisture will start condensing on the agar. And then the agar will wick it all throughout the agar. So it actually will never form a drop and will never drop down. But even if it does drop down, it drops onto the lid and there's no bacteria or colonies growing on the lid. So that's why we put everything in the incubator upside down. So when you put plates in an incubator, they should be put in the incubator upside down. Any question about that? And then something that's nice is we write on the bottom of the plate, so it's actually easier to read what you're getting out of the incubator. Bacteria colonies will grow on the agar plate, and bacteria colonies have a rather smooth textured appearance, although there can be exceptions. In comparison, fungal colonies tend to be furry or hairy. And so when you look at colonies, you can usually tell if it's a bacteria colony or a fungal colony. We do not grow fungi in our microbiology lab because fungal spores tend to go everywhere in the air, and then they can easily cause contamination. So we try to reduce the fungi in the lab. And so we don't want fungal colonies, but we do want to grow a bacteria. And it's good to be able to look at the plate and then determine whether you have fungal colonies, which are contaminants growing, or whether you have bacteria growing. And when you look at the plate, you can usually tell. Like right here, this plate colony looks fuzzy or hairy, and this one looks fuzzy or hairy right here. And this region, it looks fuzzy. These are fungal colonies, whereas this colony, which is sharp, smooth edge, sharp edge, and the same with that one and that one, these are bacteria colonies, and the same with that one. Any question about that? All right, so there's a video you can watch on binary fission, how cells divide by binary fission. Oops, let me go up. Did I talk about that? Yes, I haven't talked about this though. Each colony is made from 1 million to hundreds of millions of cells. And when we first put the bacteria on the cell, this colony may have started from a single cell. It may have started from a clump of cells, but it may have started from a single cell. And then that single cell divides by binary fission into more and more cells. And when you get about 1 million, it'll be big enough, like that cell colony there probably has 1 million cells. And these here have several million, maybe even hundreds of millions of cells, depending on how big the cells are. You can look at the colony and then describe its characteristics. You can say the colony has a shape, such as circular, rhizoid, irregular, filamentous, spindle. For shape, we're looking at the shape of the colony. For the margin, we're looking at the edge of the colony. So entire is, the margin is, I don't know, it's like a circular colony. Undulate, the margin undulates. Lobate, curled. And I'll never quiz you on what's the difference between undulate, lobate, and curled, because they're too difficult. They're too similar. And then I may ask, is this rhizoid or filamentous, or is an entire margin? But I'll never ask you, is it rhizoid or filamentous, because those two are too similar to each other. Elevation, to get the elevation, you really need to remove the lid of the plate and then look at the plate on its side, whether the colony is elevated. If not, it's flat. It can be raised like a mesa. It can be convex. Pulvinate, just extreme convex. I'll never ask you to distinguish between those two. And then umbunate is a mountain shape. You can look at the size of a colony. I'm going to start at this end. Anything that's larger than the head of the, not the head, what do you call that? The back portion of the pen, or the eraser, anything that's larger than the eraser of a pencil is large. Anything that's the size of the pencil or pen, and we're talking about a dull pencil, is small. Anything between the point of the pencil and the eraser of the pencil is moderate in size. And then anything that's a pinprick, we call punctiform. Texture, there's smooth and rough colonies. Appearance, whether it's shiny, we call that glistening or shiny, or we call it a dull colony. So let's come on up to our colonies here. Like this one is a shiny colony. That one is dull. Pigmented or non-pigmented. A pigmented colony is one that has color. So if you can see color, purple, red, yellow, it is pigmented. If it's not colored, and that could be cream, tan, white, off-white, those we call non-pigmented. And then there's optical property. Most colonies are opaque. You can't see through them. Translucent, that would be like slightly muddy water. It's translucent. You can see through it. So you can see through the colony, but it's, I don't know, you can't see perfectly through it. It's not like crystal clear water. And then there's transparent. You can see through the colony. Very few colonies are transparent. And when we come down here, most of these colonies are opaque. That one's opaque, that one's opaque. This one on the edge there is translucent. In this region, I would say it's opaque, but on the edge here, it's translucent. Any questions about any of that? And I don't have any transparent colonies. So you can use this table to give the colony characteristics. I will generally make this table available to you if I'm asking anything complex. But if I give you the colony, I should expect you to be able to say whether it's smooth or rough, shiny or dull, pigmented or non-pigmented, opaque or translucent or transparent. All right, any questions there? All right. Colony A is large, circular in its shape. You could also call that, because this isn't perfectly circular right there, you could also call that irregular. Entire edge. Pulvinate, but I can't see that. You'd have to look at it from the edge. I'm not going to go through the others. You can read through it. When we talk about colonies, you can talk about the abundance of colonies. That would be the total number of colonies on a plate. And you have to count them. That's too many to count. But if I come up to this plate here, oh, that's still too many to count. I don't know. It's about 10 there. So this would be 30 to 40 in colony abundance. And the abundance is just the colony number, however many colonies there are. Diversity is the different kinds of colonies. And then you lose the characteristics to determine how many different kinds there are. Like this colony is definitely different from those, but all of these and all of these could be the same species. So this would be 1, 2, there's 3, and that could be the same as that, but it could be different. So we'll say 3, 4, maybe 5. This one's off-white, so 7, 5, 6, sorry. I think I counted that one, and that one could be the same. So this one, and then there's that one there, that's different. So that means there's whatever number I was on, about 7. So this would be 8. And then that one could be the same as that one. So we'll leave it. So this one has about 7 different colonies on the plate. So the diversity would be about 7. Now I've got another question down here. Let me scroll to it. Looking at the cats. Come on. Sorry, my mouse is not working. Ah, come on. There we go. So what is the abundance of cats in this picture? Come on, what's the abundance of cats? Somebody has to answer.
7.
1, 2, 3, 4, 5, 6, 7. Yeah, you just count them, and there's 7 cats. And what's the diversity of cats?
3.
3. Okay, these are all cheetahs. These are all lions. So I would say 2.
Okay.
The diversity is 2. I'm not sure where you're getting the 3. You think those are 2 different species?
No.
Yeah, that's a male lion, that's a female lion. So the diversity is 2 for this one. On something like this, I would expect exact numbers, but when I'm giving you a plate like that, I don't expect exact numbers. So you can estimate the number of colonies here. And somebody want to guess? I'd say there's about 100 different colonies there. And the diversity, this is going to be tough because there's so many there. 1, 2, 3, 4, 5, 6. No, that could be the same as that, but it could be different. So there's at least 6. Actually, that small white one would be 7. So there's at least 7 in the diversity. But if you were to tell me something like 10, and I counted 7 in diversity, that's okay. But if you say the abundance is 100 and the diversity is 50, I would say the abundance is okay, but the diversity being 50 is too high. Because this one and this one, and then this one and this one, and that one, those are all the same. And then these right here are all the same too. This one, and that one, and that one, and that one. So there's many of the same species of colonies on this plate. So you can be slightly off, but don't be off by too far, or I'll say no. You got to be more exact. All right, so know the terms. I'm not going to go over those, but you should know the terms. And then complete the laboratory exercises. You can ignore the references. Oh, here we go, the answers there. Abundance 7, hey, that's what I got. And then 2 lions and cheetahs, colonies, so 80 to 90. Of course, I said around 100. And diversity at least 10. And I said at least 7. All right, let's go on to the worksheet. So type your answers in this worksheet or print it out and scan it and send it to me. We're talking about the ubiquity of organisms. Answer the question, what's in the air you breathe? You don't need to answer this question because this is part of the procedure, but you should answer everything that's got a blank or a question mark. And that has a question mark, I know, but you don't need to answer the procedure one. Just telling you, answering what's on the procedure. So you need to tell me the colony abundance here, the colony diversity, and then give me what steps you could do to reduce airborne contamination of this plate. So you're working on an agar plate. What steps could you do to reduce airborne contamination when you're working on the plate? Procedure 2, what gets on your arms when you put your arms on the bench? So read through that. And then you're disinfecting the table and it's asking, did disinfection work? There's the plates with the colonies before disinfection and the plates of colonies after disinfection. So answer, did disinfection work? And then answer, what is the purpose of allowing the table disinfectant to air dry? People sometimes have difficulty with that question. Procedure 3, what's on your hands? You're going to be looking at your hands, the microbes on it before you wash your hands, and then after you wash your hands. And the first question, does hand washing work? Answer that question. Suggest one reason why hand washing did not result in the complete removal of microbes from your hand. Like there's some microbes on your hands after you're washed. If you need to go online, you can answer that. You could, if you know the answer, you could answer it. I don't think that's discussed in the lab module. It has something to do with the normal microbiota on your skin. And if you get your answer doing your research, you should give a citation of your information source. Procedure 4, when you kiss your significant other, what are you giving him or her? Meaning what's on your tongue and your lips? So it looks like that's French kissing. When you look at this picture, this is the microbes from somebody's lips, the bottom lip and the top lip. And this is the microbe from a tongue. They press their tongue against the plate here. And they must have a long tongue or they really put out their tongue far. When you look at this picture, I've got it here. Oh, there it is there, the note that the smear appears from the tongue. If you look at that closely, you'll see that this smear here is made up of lots of colonies right here. Okay, so these colored ones are not the only colonies. There's a bunch of colonies right here. So that the colonies are so numerous, the growth is almost confluent. But each of these, if you look closely, those are colonies in there. And then answer the questions. Which place has higher colony abundance, the lips or the tongue? Why do you think this is the case? And then it goes through the procedure that we grow the tongue plate at a higher temperature than the lip plate. And why do you think we're doing that? Why is the tongue plate grown at a higher temperature than the lip plate? Answer that question. Procedure 5, are there microorganisms in the soil? What you do is you take out a little bit of soil, put it in a liquid, scatter the liquid on the plate, and then allow it to grow. And you can't do this, so we're providing pictures for all of these exercises. And here's the picture of the soil plate, and it asks, what is the colony abundance? Is it possible to accurately answer this question? Explain your answer. And is there greater colony abundance in the soil compared to the lip plate? Shown here. And then when you're done, submit your answers to Lab 01. It is due at 11:59 p.m. Saturday. Let me remind you that Lab 00 must be submitted by 8 a.m. Friday morning. And if you do not submit it, then I will drop you from the class. Any questions? If not, go ahead and get started on the lab. I will be here until 8 o'clock to answer questions. I may be starting my dinner, in which case I'll be away from the computer for a little bit, but I'll come back after a few minutes. All right, any questions?